Hi guys, Miss Francis here. Today we're going to talk about eukaryotic cells. As mentioned earlier, we know that eukaryotic cells are compartmentalized, and what this does is it creates smaller environments for that compartmentalized organelle to do its specific function. Now, how is this organelle compartmentalized? Well, it's got a double layer of phospholipids, which gives it some protection and separation from the other organelles. So, for example, the membranes of the mitochondria enable that mitochondria to function specifically during cellular respiration. So what they do is set up this extensive and elaborate system of internal membranes, and that partitions the cell into compartments. The membranes also participate in metabolism, as many enzymes are built into the enzymes, and the barriers that are created by the membranes of these different compartmentalized organelles provide different local environments that facilitate the specific metabolic functions associated with that specific organelle. So just to summarize, eukaryotes have compartmentalized cell structure, which enables the efficiency of metabolism and localized conditions. Eukaryotes fall into 4 different groups. You've got your fungi, protists, animals, and plants. From living environment, you should remember that there are some distinguishing features between plant and animal cells. So if you click here, you'll get to Cell Alive Interactive, and you can test yourself as far as how much you remember from plant cells versus animal cells. So here we have an animal cell. You notice it's circular in structure, and an animal cell is going to have some features that won't be found in a plant cell, and a plant cell is going to have some features that won't be found in an animal cell. First of all, look at the shapes. Animal cells are much more rectangular than plant cells. Animal cells will not have chloroplasts. They won't have a central vacuole. They won't have a cell wall. They will have vacuoles. They just won't have a large permanent central vacuole. Whereas if I look at the plant cells will not have lysosomes. They won't have centrioles. So lysosomes are used in digestion. Centrioles are used during cell replication, and flagella are found in some plant sperm. So here is another general picture showing some differences, distinguishing features between plant cells and animal cells. And here is an overall chart showing some differences. If you click here, you can watch a Stephanie Castle video about the differences between plant and animal cells. So you will have to label eukaryotic cells, and the picture that you might have to label could be an artist's rendition, or it could be a micrograph like you're seeing here. And you'll need to be able to identify the major organelles, such as the plasma membrane, rough ER, mitochondria, vacuole, Golgi apparatus, the nucleus, and the nuclear envelope. Furthermore, you could be asked to draw a picture of a eukaryotic gland cell. For example, they might say draw a liver cell or draw a pancreas cell, and you're expected to know that that means it's a eukaryotic cell, and you would have to draw it. Stephanie Castle gives us a wonderful tutorial on how to draw the eukaryotic cell. You will need to be able to identify whether or not you've got a plant cell or an animal cell looking at micrographs. So here you've got a plant cell, and you can tell because it's got the cell wall here, it's got a central vacuole here, and these are your chloroplasts over here. All right, that's it for today. The next time I make a film, it's going to be talking about the endomembrane system. So I'll talk to you then.